When a human body drops just a few degrees below normal, the blood begins to turn against itself. Severe hypothermia triggers our platelets to activate and clump together, forming lethal clots that block circulation and destroy tissue. But the European ground squirrel routinely survives a winter hibernation, turn into a lethal traffic jam of clots and trigger massive organ failure. Yet they wake up perfectly fine, without a single sign of ischemic injury. The squirrel manages this through a rapid physiological shift. As they enter torpor, they strip away 90% of their circulating white blood cells and platelets, clearing the bloodstream of cells that cause damage. They don't survive the freezing temperatures by fighting the cold. They survive by temporarily dismantling the specific parts of their immune system that the cold could weaponize against them. Researchers tracking this torpor cycle notice something highly unusual happening at the 24-hour mark. This chart shows blood cell concentration. The tall left bars are the summer baseline, plummeting 90% during torpor. The mystery happens during arousal. At the 1.5-hour column, cells return to summer levels, even though the inset graph shows body temperature hasn't recovered. Recovering millions of complex cells in 90 minutes presents a problem. According to everything we know about how long it takes cells to grow and mature, that speed is a biological impossibility. So where do all those cells go for the winter? The first logical assumption is that the cold kills them off in a process of mass cellular death, known as apoptosis. To test that, researchers looked at the squirrel's spleen, which acts as the body's filtration system for processing dead blood cells. If 90% of blood cells died all at once, the spleen would work overtime, and a region called the white pulp would swell. But the data shows the white pulp remains stable at roughly 4.3%, whether the squirrel is warm and active or frozen and asleep. If the cells aren't dying, the other standard explanation is that the squirrel's body is manufacturing millions of new cells the moment it wakes up. This timeline chart compares the squirrel's recovery window to standard biological production rates. Generating new lymphocytes, like T cells, physically requires weeks or even months of biological lead time. Getting it done in 90 minutes simply cannot happen. By process of elimination, the data suggests a different reality. The cells aren't dying off, and they aren't being rapidly rebuilt. They are being temporarily docked inside peripheral tissues and bone marrow. Instead of destroying and recreating its immune system, the squirrel uses a reversible storage system. It packs its most volatile cells away, ready to be driven back into the bloodstream the moment things warm up. These dynamics provide a natural model for addressing the tissue damage caused by trauma and ischemia. If researchers can reverse engineer the exact biochemical signals the squirrel uses, we might be able to manually trigger this hide and return response in human patients. Doctors could potentially bench a patient's white blood cells to prevent the organ injury that occurs during severe sepsis. Surgeons could drop a patient's body temperature during complex procedures without triggering lethal cold-induced blood clots. The biological pathways for managing these traumas are already functioning in the wild. Evolution has already perfected them. They're just sleeping quietly in a burrow, right beneath our feet. This exploration into the extremes of biology was made possible by Synestex Systems LLC and founder Shane Persier. If you want to support more deep dives into the science shaping our future, please like this video and subscribe to the channel.